Hey everyone, Mr. Larson here. So we've now gone through basically what DNA replication is, how it's replicated. We went through transcription and how a DNA molecule becomes a messenger RNA. And then we went through the process of translation, where that messenger RNA eventually becomes a polypeptide strand and eventually a protein. So now we're going to talk about what mutations are. We've talked about it a little bit in our the cancer digital lesson, but what really happens if things go wrong? If there's a mistake in the DNA molecule, if there's a mistake in the RNA molecule, like what could potentially happen? So here we have a couple specific types of gene mutations. One is called a point mutation. So that involves a change in one or only a few of the nucleotides because they occur. So basically, this is just what happens, a sort of mutation that only happens with one or maybe two or three different nucleotides within a DNA sequence. We have what are called frame shift mutations. So basically, what that means is a nucleotide is deleted from a sequence accidentally. This sometimes happens. And then what is read is completely different than what was read before. So let's say you have the phrase Taco Bell. So here we have Taco Bell. So a point or frame shift mutation, basically, let's say our A just disappeared. It's gone. There's no more A in Taco Bell. Now when you read it, it just says Taco Bell. It doesn't make sense. You can't read it that way. Same thing with frame shift mutations. This is a very similar thing because now it's being read differently because there's a complete letter that has just disappeared. Here are a couple other examples. We also have what are called substitutions. So let's have our example here. Taco Bell again. Taco Bell. So for substitution, let's say this O, instead of an O, it's an E, is now called Taste Bell. Doesn't make sense. That is basically a substitution, giving it a completely different meaning that nobody truly understands. And then even when it is transcribed, that becomes a problem as it moves forward. Now, insertion. Let's go back to our idea of Taco Bell. Now, let's say instead of it being Taco Bell, we have an insertion where we actually add a T here. Now it's called Tacto Bell. That doesn't make any sense when you read it, and it causes problems along the line as it's read. And you also have the deletion, which is sort of the one that I mentioned earlier. So each of these represents a different type of gene mutation. Here you can see, instead of my silly example behind me, with actual DNA, RNA, and amino acids. So this is the normal one that you see here, which creates met, arg, thur, and leucine. Here you have a substitution where it actually adds a different, or changes this G, or this C here, this C here to a T, which changes the meaning of this codon, which now it is now histine instead of an arginine. Now insertion. So an insertion basically means it inserts a new letter. So it now has a T in it. So with this T, instead of it being methionine, like it should be, like it was above, we're now in isoleucine, which is a problem. Because basically what that means is it's could potentially change the entire protein. Could change the function of the protein, which in the long run really causes a problem. So each of these mutations could cause a problem sometimes, and sometimes we're just lucky where it happens and nothing really changes. So there's also such things as chromosomal mutations. So now instead of there being a frame shift mutation where there's only a single DNA nucleotide, you might have a mutation in the whole chromosome in general. which could be really, really bad and can lead to serious health effects. So during cell division, a chromosome may break or lose a part of it, which is a deletion of an entire portion of your genetic material. So it could be a really big problem. A duplication is where a chromosome breaks and it actually attaches to a different chromosome, giving it twice the amount of genetic material, which could potentially be a problem if it creates a new cell. So you don't have let's say you have two over here, and then one broke off and joined this one. So instead of it being even, you now have more genetic material over here and less genetic material over here. So the cell with less genetic material is going to have issues potentially creating different proteins, while this cell, which has more genetic material, is going to have issues creating too much of those proteins because it has so much genetic material. Now, an inversion is when a chromosome breaks and reattaches in the opposite direction. So let's say we have our marker here. It breaks off this top and attaches here at the bottom. It's going to have different properties than it did before. So just like now I can write with it if it has it like this, where if it has the top on, I won't be able to write with it. Basically, it has different properties because of that sort of change. A translocation is where a chromosome breaks and attaches to a different chromosome. So a non-homologous chromosome basically means that it is not the same. So non-homologous, basically just referring to the fact that it is a completely different chromosome, meaning that information is in a completely different section of the cell and could lead to issues down the line. So each of these just represents the different types of mutations. So here you have a deletion, so you completely lose that B. Here you have an insertion where you actually double the amount of information for B. Here you have translocation, which is a completely different, creates a completely different sequence. It can kind of throw it all off balance. And this could all lead to various different problems along the line. So that is all the mutations. If you have any questions, feel free to ask and continue on with the digital lesson.